Hello, my name is Amit Toprak and I'll be explaining the endothelial tip cell paper today. So the endothelial tip cells, they differentiate via various cell signaling proteins. And one such example is TGF-beta. And TGF-beta actually binds to ALK5 receptors in order to initiate a whole bunch of mechanisms. And basically the significance of the whole paper in the first place is to study the angiogenesis and capillary branching mechanisms in humans. And this is important because we can, once we understand it, treat vascular diseases. And one such example that's clarified in the paper is oxygen-induced retinopathy. In humans, I believe this is more clearly characterized as retinopathy of prematurity, ROP. And so in order to investigate the mechanisms of angiogenesis and tip cell differentiation, this paper outlines a mouse retina experiment. And the experiment starts out with the single cell RNA sequencing of a retinal sample. And the single cell RNA sequencing is first accomplished by magnetic activated cell sorting, which is it's an enrichment process in which cells of interest bind with high affinity molecules, and then an electromagnetic field pushes them to one side of a tube, let's say, and that's the method in which these cells are isolated. And so endothelial tip cells or endothelial cells in general were actually isolated from a retinal tissue. And after that, with single cell RNA sequencing, there were a couple quantitative methods employed in order to analyze all the relevant data. So expression levels in different epithelial tip cells were investigated, and furthermore, they were made more accessible to the reader through UMAP plotting, which I believe is uniform manifold analysis projection or something like that. But basically, you take a whole bunch of proteins or RNAs or genes, let's say, and you analyze them through a whole bunch of dimensions. And this could be things like size or transcriptome similarity, et cetera, et cetera. And then you compress it into two dimensions, UMAP1 and UMAP2, and that's the X and Y axis. And so however close two clusters are to each other on the x-axis or on the y-axis, that's how closely they're related from a genotypic or phenotypic standpoint. Alongside quantitative measuring, there were also qualitative measures employed. One such example was fluorescent microscopy. And basically fluorescent microscopy used cell markers specific to the cells of interest. So this could be of epithelial cells or proliferative cells, or even D cells or anything like that. And we basically saw through both the qualitative and quantitative measurements that P6 and P10, that is postnatal day 6 and postnatal day 10 cells, were quite different in their distribution of endothelial cell types. And first, the paper starts out with establishing differences between P6 and P10 and all types of endothelial tip cells. And then, in order to investigate the role of TGF-beta signaling, the authors of the paper start what I've labeled as the ALK5I eco mice experiment, which is basically the exon 3 deletion in the ALK5 gene. And this basically is the way that this is generated is through crossing CRE mice with LOXP mice, also known as flox mice. And then that basically creates a lineage cross called CD5 CRE-ERT2. The CDH5 CRE-ERT2 mice, when you administer tamoxifen to them, what happens is the exon 3 deletion becomes induced. And so what happens is these cells, these endothelial cells, of this strain, once tamoxifen is administered, they are lacking the ALK5 receptor or gene even. So what happens as a result of this is all the downstream processes become deleted. One such example, the canonical TGF-beta pathway is completely inhibited. So we saw this through the Western blot, where there was like a 65 kilodalton strip of protein missing, and that was the PSMAD 2 and 3, and they were completely absent from the Western blot. So PSMAD2 and PSMAD3, they phosphorylate the downstream transcriptional factor called PSMAD4. Of course, P stands for phosphorylated, so actually phosphorylate SMAD4 to turn to PSMAD4. So once SMAD4 is phosphorylated, it initiates a whole bunch of genetic phenomena, such as epithelial mesenchymal transition (EMT), proliferation, apoptosis, inflammation, and cell cycle arrest. Of course, in a vacuum, if you have a cell in cycle arrest for long enough, then what's going to happen, as a result is apoptosis. And this is just one of those things that's present in a whole lot of pathways, like the MAV-S pathway. I believe that's initiated by toll-like receptors, but that's an immunology thing. It's not very relevant. So the canonical TGF-beta pathway is completely inhibited, and what else happens upon tamoxifen administration is that Rho kinase A, that doesn't get phosphorylated. So as a consequence, ROC1, ROC2, they don't get phosphorylated, they don't get activated, they don't activate their downstream protein cofilin, and cofilin binds to non-muscular actin and is involved in the regulation of actin polymerization, and also in order to get the non-canonical pathway to activate cofilin, you also need the co-activation of the cytokine, and that's something that I did. That's some research that wasn't included in the paper, but I found on the internet anyways. I thought that was pretty cool. So the cofilin, once activated, it regulates cell assembly dynamics. And let's say if you're an endothelial cell and you're becoming a tumor, some carcinogenic processes are happening. What ends up happening when you overexpress cofilin is that metastasis can happen. So at the end of the day, what ends up becoming clear is that TGF-beta is an immensely important molecule in the differentiation of endothelial tip cells. And without this, retinopathy can occur, and the retina isn't formed well enough. Other molecules, or other hormones can't really rescue this. If you overexpress WNC, which guides diving tip cells, you still have pathogenesis occurring. And of course, if you don't have diving tip cells, then you can't establish the blood-retinal barrier. Your visual function is completely out of whack. Your neuronal activity is out of whack. A lot of the molecules associated with superficial tip cells, they become expressed more prominently than if you were to have diving tip cells. So what ends up happening is there's a complete undifferentiation. There's an increase in proliferative tip cells or proliferative endothelial cells, and you don't get this very key player diving tip cells in the whole mechanism of the retina.